Good afternoon, everybody. We're just going to give everybody a moment to join, and then we'll get started momentarily. We have a full day, and we want to use every minute that we have. Perfect. As I said, we do have a full day, so I want to get started as quickly as possible. So first and foremost, welcome. Today is the first general session of the Newborn Screening Bootcamp, and we're really excited for this year's bootcamp and the agenda that we have. In addition, I want to thank Baby's First Test. This is the third time that we have co-hosted and co-run this event with them, and they continue to be wonderful partners, not only for the bootcamp, but for newborn screening efforts in general. So I want to send a big thank you to Baby's First Test and their team for all their work today, next couple of weeks, and for helping to prepare us for this event. And I also want to put a thank you into our sponsors, who without them today would not be possible. I just want to do a little bit of background on the EveryLife Foundation for those who are unaware of who we are and what we do. So the EveryLife Foundation for Rare Diseases is a nonprofit, nonpartisan organization dedicated to empowering the rare disease patient community to advocate for policies that advance the equitable development of and access to life-saving diagnoses, treatments, and cures. And what we do is, again, we do not speak for the patients. We provide training and education, such as today, to make their voices heard, because we're well aware that by activating patient advocate, we can change public policy and save lives. Because as somebody who works in DC, speaking with policymakers all the time, they know what I'm going to tell them, but hearing from their constituents and from their advocates about the personal stories is really impactful. And so we want to jump right into today. We're really excited for today's session. It's going to be a wonderful discussion about the RUSP process and the RUSP review process, and we'll get into a little more of that as we get forward. And so we're going to originally get a presentation from Dr. Cynthia Powell, who is the chairperson of the Advisory Committee for Hereditary Disorders in Children and Newborns. And then we'll move into a panel after that to help to kind of work through the presentation and answer any questions.
Without further ado, I'm going to introduce Dr. Powell. And I have taken the liberty to pull highlights from the bios of our panelists today, because we would spend the next hour and a half sharing everything that they've accomplished. So we're really grateful to have all of you here with us today. So Dr. Powell, as we mentioned previously, is currently the chairperson of the Advisory Committee for Heritable Disorders in Children or Newborns and Children. She's a professor of pediatrics and genetics at the University of North Carolina at Chapel Hill School of Medicine, where she sees patients, teaches students, residents, and fellows, and participates in research. She's a board-certified clinical geneticist, cytogeneticist, pediatrician, and genetic counselor. She did her pediatric residency here in DC at Children's National Medical Center and medical genetics fellowship at Children's here in DC and the NIH. She's past president of the Association of Professors of Human and Medical Genetics, chair of the U.S. Federal Advisory Committee on Heritable Disorders in Newborns and Children, and past president of the American Board of Medical Genetics. She's a member of the board of directors of the American College of Medical Genetics and Genomics, serves on the North Carolina Newborn Screening Advisory Committee and the North Carolina Genetics and Genomics Advisory Committee. Her research interests include newborn screening, genomics, birth defects, and genetic syndromes. She led the North Carolina Newborn Exome Sequencing for Universal Screening Project, a 5-year project funded by NIH investigating the utility of next-generation sequencing in newborns. So you can see why we're really delighted to have Dr. Powell here. She's also a longtime unwavering advocate for our patient communities that are working in the newborn screening space. So Dr. Powell.
Thanks very much, Annie. And thank you to the EveryLife Foundation and Baby's First Test for the invitation to join you today. It's a pleasure to be here. Next slide, please. So I wanted to give a brief overview of the Advisory Committee on Heritable Disorders in Newborns and Children, how it got started, how things currently work, and then go on to what are some of the proposed changes that are being considered. The Advisory Committee was established in 2003. And that was at the time when there was a large discrepancy in the number of conditions that states were screening for in newborns, with some having gone already to the use of tandem mass spec, which greatly increased the number of conditions, whereas other states were still only screening for maybe 5, 6, or 7 conditions. So an advisory panel, expert panel that was funded by HRSA, and was led by Dr. Rodney Howell and Dr. Michael Watson, representing the American College of Medical Genetics, and included experts in newborn screening types of conditions. And they established a core panel of 29 conditions and 29 secondary conditions, meaning those that would be picked up in screening for the core panel of conditions. The secretary approved that panel, and at that time the RUSP, the Recommended Uniform Screening Panel, was established in 2010. The committee has been funded, along with many other things, through the Newborn Screening Saves Lives Act, which was enacted in 2007, began in 2008. Since the committee has been established, additional conditions have been added that are severe combined immunodeficiency, critical congenital heart disease, Pompe disease, Hurler syndrome, X-linked adrenoleukodystrophy, and the last condition added to the panel was spinal muscular atrophy. Unfortunately, in 2019, although the House passed the reauthorization for the Newborn Screening Saves Lives Act, the Senate did not pass. So we had to sort of reform the committee, and it's now essentially a discretionary advisory committee that is still able to work, but is not able to do all the things that we'd like to because of the failure of the Newborn Screening Saves Lives Act to be reauthorized. Next slide, please. So some of the essentials regarding newborn screening and how the committee really was formed and initially decided all the different parameters to use and thinking about new conditions to add to the RUSP were based on several reports ranging from the World Health Organization in 1968 to the ACMG panel that was published in Genetics and Medicine Toward a Uniform Screening Panel is the title of that, and that was in 2006. Next slide, please. So the report from the National Research Council stated that mandated screening could be justified only if there was evidence that it would prevent death or other serious harm to the affected individual, and that procedural guidance must exist in order to maximize the effectiveness of genetic screening programs. Next slide. The Institute of Medicine report stated that as basic principles to govern newborn screening, the committee recommends that such screening take place only when there is a clear indication of benefit to the newborn, that a system is in place to confirm the diagnosis, and that treatment and follow-up are available for affected newborns. Next slide. And currently there are 35 core conditions on the RUSP and 26 secondary conditions. And those you can find on the HRSA committee, ACT-DNC committee website. Next slide. So the RUSP is a list of conditions recommended by the Secretary of Health and Human Services for states to screen for as part of their state universal newborn screening programs. Conditions on the RUSP are chosen based on evidence that supports the potential net benefit of screening, and we'll have a chance to talk more about that later, the ability of states to screen for the disorder, and the availability of effective treatments. It's recommended that every newborn be screened for all conditions on the RUSP. Next slide. Most states screen for the majority of disorders on the RUSP. Newer conditions are still in the process of adoption, but it's really left up to individual states as to what conditions they will screen for. Some states screen for additional conditions that are not on the RUSP. And although states ultimately determine what conditions their newborn screening program will screen for, the RUSP establishes a standardized list of disorders that have been supported by the committee and the Secretary of HHS. Next slide. And this is a basic overview of how the process works. Once a condition is submitted, a nomination package is submitted to HRSA for review. HRSA sometimes sends it back because it's incomplete. There's certain things missing. So they'll send it back. Then the nomination package can be resubmitted. After HRSA goes through and sees that all the elements are there, then there's what's called the Nomination and Prioritization Work Group, which is made up of members of the full committee. I think 5 members from the full committee, and they review the package, and then they present it to the full committee that then votes. And either the committee votes to move it forward for a full evidence-based review, or they vote that it's not ready for full evidence review and send it back, providing guidance to those who have submitted the nomination to let them know how the nomination can be strengthened. If it is voted through, then an outside group, currently headed by Dr. Alex Kemper, who runs the Evidence Review Work Group, and they have 9 months to do a full evidence-based review of that condition. Their results, findings are then presented to the full committee that then votes whether to approve the condition for the RUSP or not approve, in which case, again, the group that's given the nomination is provided feedback as to how the nomination can be strengthened. If it's approved by the committee, then it goes to the Secretary of Health and Human Services, who may request additional information or approve it. And then if the Secretary approves it, it would then be added to the RUSP. Next slide, please. So for the condition review process, a group that is considering putting forth a nomination package should form a multidisciplinary team. The team should include researchers and or clinicians with expertise on the condition being nominated, advocacy and or professional organizations with knowledge of the issues relevant to newborn screening, and interested consumers and individuals. Next slide. As I mentioned, then the Nomination and Prioritization Work Group looks at the package that should include a cover letter, letters of support from as many individuals as would be pertinent. There's disclosure forms that have to be completed, responses to the nomination form, specific questions that are asked on the form, and then supporting data and scientific clinical references to substantiate all the responses to the nomination form questions. And the current nomination form is found at the link provided there. Next slide. So in order to submit a nomination, the requirements are that there must be a validated laboratory test, widely available confirmatory testing with a sensitive and specific diagnostic test, and a prospective population-based pilot study. Next slide. As I said, HRSA then reviews the package for completeness, and again, the Nomination and Prioritization Work Group goes before the committee to present, or I'm sorry, goes before the committee to present their findings, and then the committee votes on whether or not to move it forward for evidence-based review. Next slide. The external condition review work group completes a systematic evidence-based review, provides updates to the committee, and presents a final report to the committee on the assigned condition. The committee deliberates and votes on the condition, and the committee uses a decision matrix to guide their final decision. And as I mentioned, the secretary makes the final decision on whether or not to add or not the condition to the RUSP. Next slide. Basically, the highlights of the matrix that's used are indicated here. First of all, whether there's net benefit to the population of newborns screened. Is there a high certainty of that net benefit, or a moderate certainty, or higher moderate certainty that the targeted condition would lead to small or zero net benefit? And then are states capable to offer this newborn screening? Are state labs and the whole state newborn screening system, is it ready to add this condition to their state panel? Next slide, please. And this is what the decision matrix looks like. It's a bit complicated. I won't go through all of it, but certainly available there. But essentially, those are the highlights: the readiness and the net benefit. Is there net benefit, and what's the certainty of that net benefit? Next slide, please. So now to go on to why we have been considering changes to this process. In February, HRSA convened an expert advisory panel to address the process through which a condition is considered for or included in the RUSP. It was felt that it would be important to inform the committee about ways to strengthen the evidence review and decision-making process, since the current process had been in place for several years already. And also, it was recognized that we needed to develop consumer-friendly guidance to enhance the transparency about the process and any changes. Next slide. So the areas that were focused on included the nomination form itself and the process, the evidence-based review, the decision matrix, and what I'm not going to talk about today was including a review of conditions that are already on the RUSP. So something that we have not been doing, but was under consideration. Next slide. So the guiding questions were: what issues or changes are needed in the committee process? What are the next steps needed? How can we address these issues? Which actions can be done immediately, and which would need further discussion, additional research, or changes in policy? Next slide. And this was the timeline beginning in February of 2019 up until our last meeting in August, where the final report was presented to the committee, and the committee was given a summary of proposed next steps, and the committee will vote on these changes at our next meeting in November. Next slide. So here's a summary of the issues that were identified. Next slide. So in terms of revisiting the nomination process, the issue was guidance about the process from HRSA or the committee is limited about the process itself or specific criteria considered for nominations. The actionability is that consumer guidance and nomination frequently asked questions will be posted or developed and posted on the committee's website in the coming fiscal year. The next issue was that information requested from the nomination form does not directly link to specific and relevant information needed for the evidence review in areas such as registries, unpublished evidence, screening algorithm and resources, long-term follow-up, etc. And again, revisions proposed for the nomination form are in place, and the new nomination form will be posted in fiscal year 22. Next slide. The issue being that no systematic review or landscape scan is given in the nomination package. The information that the nomination and prioritization work group has to go on could potentially be biased, not so much intentionally, but the fact that, you know, those who are putting in a nomination package are generally folks who are, you know, very favorable to adding their condition to the RUSP. Given the limited time that we have for these evidence reviews, there was discussion and recommendation they have a scoping review that the NNP work group would give. However, that would require development of new procedures. It would be quite time intensive, and no committee action was taken at that time. It was felt that there would be further discussion about that, having this scoping review of a condition before it's presented to the committee and before it's decided whether or not to go forward with the full in-depth evidence review. Next slide. Assessing the published evidence. So this is a real challenge for extremely rare conditions, often where there isn't much published information available. The evidence review group does utilize what's called gray information that might be an abstract or a poster presentation at a national meeting, but something that has not undergone rigorous peer review and published in the medical literature. So the problems related to some of that include that the case definition is not always clear. Like, what is the condition that newborn screening would be intended to detect? The case definition should specify screening targets and be cognizant of the screening procedures, and that changes in the case definition based on evidence should be raised early in the review and presented to the committee. Also, that critical outcomes are dependent on the availability of evidence. So what are the critical outcomes that should be assessed and considered in terms of the net benefit of doing newborn screening for this condition? Also that the patient or family or other values were not always being considered, and information was not available on them. So the committee processes in fiscal year 22 will be adopted with critical outcomes identified up front. And there was further discussion about ranking stakeholder importance, about how rankings could be gathered, whose rankings, looking at the input from the committee, family, general public, and other stakeholders in terms of their thoughts about newborn screening for a condition. Next slide. Also assessing the unpublished evidence. There are things that are ready for implementation. The review group will expand their current procedures for assessing the gray literature, incorporating other methods available to use some of the expert-derived evidence that may be unpublished once relevant meeting abstracts or other unpublished resources are identified. And then looking at additional data such as data in registries, and hopefully we'll have more time to discuss that in the discussion part of this meeting. Next slide, please. Thinking about what are the costs of implementing a new condition, in terms of how states can be assessed for their readiness to begin a new condition. Some new steps are being incorporated into the survey. There's a new survey form that's been developed. The first time it will be used will be in the review process for MPS II, Hunter syndrome. and hopefully that will gather additional information that will be valuable in terms of the public health system impact. Next slide. And revisiting the decision matrix, no major changes are being made. However, there have been discussions about the B rating where there's moderate certainty of net benefit, but there may be not a lot of information available at that time. And we plan to have further discussions about that to develop, maybe break down additionally the B ratings. Next slide. And then again, some of the other discussion was about the yes or no in the decision matrix. You know, some of the recent conditions have had some uncertainty. We felt that further research was needed. However, not wanting to delay adding a condition for what could be, you know, 2 or 3 more years. And so we're still thinking about ways to further sort of think about this. It was the committee has voted no to adding to the RUSP and provided further requirements, such as initially when SCID was reviewed and the first review for GAMT, and that there may not be any changes needed. However, that I think will still warrant additional discussion in the future. Another problem certainly is we see many more treatments under development for rare disorders, that the process has been very slow. That's one reason initially the 9-month period for the evidence-based review was incorporated into the process, was to speed things up. However, we understand that it's still a very slow process. And so future reviews might include considering closely related conditions together, such as in a panel, such as for some of the additional lysosomal storage disorders or intellectual disabilities, instead of one condition at a time. No formal action has been taken in this regard yet. Next slide. And then how to incorporate values assessment. Again, something that many of us feel is very important. However, again, it's time consuming, expensive, challenging to do this. Assessing public perspectives, patient and family perspectives. They're, you know, often very individualized, not easily generalizable. And how could we have something in place that could be utilized, such as consumers who are informed about newborn screening already, who could be asked to weigh in about specific conditions. And hopefully there will be more steps taken in the future about this, but at this time no action has been taken, but we hope to have further discussions, further research, and considering how to do this. Next slide. And then for some of the priorities for research and development activities, many of the issues, as I said, will deserve more research and planning, and the priorities will be determined, such as conditions potentially appropriate for newborn screening, and then possibly including the values assessment. So more to come in the future. Next slide. Some of the key issues, I'm not going to go through each one because there's a lot of repetition, but in the red are some of the highlighted things in terms of the nomination package, including specific case definitions for the screening target, including as best as available information is there, what is the incidence in the US for the condition, what phenotypes would be detected, what is the distribution of severity for the known phenotypes, and describe the clinical care required. I think what's often helpful would be in the form of an algorithm where if one were to screen 100,000 newborns, how many approximately would have positive screens? How would those infants need to be assessed in terms of confirmatory testing. Where would they need to go for that? How far would they need to go? What are the current treatments available, the current standard of care? Next slide. What are the clinical indications for treatment? How urgent is that treatment? Is the condition where that baby should be seen, within 24 hours of the state lab reporting out the results, or is it something that, it would be okay to wait a little bit longer? What are some of the contraindications for treatment initiation? And then, is treatment and follow-up available in most hospitals? Would one have to go to a major medical center? Are those treatment centers available? Next slide. What is the modality of the screening sample available? Is there high throughput? Is there, good sensitivity and specificity? Is there need for a second tier type of test? And then what types of validation measures are there? What are the confirmatory testing methods? Are they quantitative or qualitative? Next slide. What is the status of the confirmatory testing? Is there an FDA approved test? Are there pilot studies that have already been done? What were the outcomes of those pilot studies, or any states currently screening for that condition? What has the outcome been of those who were screened positive versus those patients who were diagnosed later on because of their clinical presentation? What would be some of the results, as I said, in states that are already doing this? Is there information from existing registries that could be gathered? And I think we'll try to talk a little bit about that later on. Next slide. And that's it. So sorry to rush through that. I wanted to make sure we had plenty of time for discussion, and I'm happy to take questions during the discussion panel.
Wonderful. Thank you. First of all, Dr. Powell, for all that information, and also to the team at HRSA who helped compile all those slides and all that information. Before we move to the panel, just one question that I'm sure is on the minds of some of the people participating, me included. If you're a part of a community that is currently working on developing evidence and compiling a nomination, what are the implications for the new forms that are coming FY 2022 if you're working off of the current forms? What's your guidance to those communities?
Right, so I think the new form will not have to be utilized until next year sometime, and HRSA will give out plenty of notice about that. So currently we're still using the current form.
Okay, great. So let's dig in a little bit further. And so for everyone, we're going to move into a panel discussion and dig in a little bit further to many of these topics. I'm going to introduce all of our co-panelists. We are going to take questions in the chat, and we have some of our team members at Every Life who are going to try and keep up with some of the questions. But we do have some topics that the panel has sort of flagged that we think would be beneficial to dig into. So we're going to try and keep up with the chat, but we do want to dig into some topics in particular. If we don't get to everything that's in the chat or all of the questions, we will try and get those questions answered and then post them later as a part of the bootcamp follow-up. So just so you know. So let me introduce everybody who's here because we're really grateful to be joined by Dr. KK Lamb and Anna Maria Serenin also. So Dr. Lamb, I'm going to just do some quick introductions of everybody. So Dr. Lamb is a research child and school psychologist who served as the project director for the Newborn Screening Evidence Review Group, or ERG, since 2011. In her role with the Newborn Screening ERG, Dr. Lamb works with the scientific lead and ERG chair, Dr. Alex Kemper, whom many of us also know, to conduct comprehensive evidence-based reviews of expanded newborn screening for Pompe disease, MPS 1, X-linked ALD, and SMA, and led development of consumer-friendly summaries of these reviews, which we all appreciate very much. She's also assisted with analysis and revision of the review procedures for the Advisory Committee on Heritable Disorders in Newborns and Children to incorporate evidence about population level and public health system impact on newborn screening, as well as other policy reports requested by the committee. And we're going to talk about some of those efforts today. In addition to our work with newborn screening, Dr. Lamb serves as Operations Director for the CTSI Special Populations Corps, which facilitates inclusion of special populations in clinical research. So then I'm going to move to Anna Marie's introduction. And I have pulled highlights, but her bio is posted on our website, and I encourage everyone to go see it because many of us are also patient advocates. And I think her bio really speaks to her commitment to just serving the broader global community. So I'm going to just share some highlights from Anna Marie's work. She's a Humphrey Policy Fellow and co-founder of both the Newborn Foundation and Bloom Standard, a social impact innovation lab developing medical resources for children in resource-poor settings. The Newborn Foundation was founded after her own baby was diagnosed with critical congenital heart defects and is focused on developing policies, programs, and technologies to improve early diagnosis, health outcomes, and access to care for mothers and babies. Under her leadership, the organization has been recognized for spearheading the U.S. effort to become the first nation to implement universal newborn screening for heart defects, the most common and deadly birth defect. Anna Marie was appointed to the Federal Advisory Committee on Heritable Disorders in Newborns and Children and serves on the Minnesota Department of Health's Newborn Screening Advisory Committee. She's drafted more than 40 pieces of health legislation, authored more than 200 policy briefings, and authored numerous manuscripts on the importance of technology in advancing early detection and treatment of neonatal and pediatric health conditions. She serves on 2 active UN working groups and was among the first formal commitment makers to the UN Secretary General's Every Woman, Every Child initiative. She serves on the Every Breath Counts Coalition, the WHO Medical Device Technical Series Work Group, the WHO Research Network on COVID-19 in Children, and is co-chair of the Newborn Foundation Research Collaborative. She's the recipient of the Betty Hubbard Maternal and Child Health Leadership Award. She holds a degree in economics from the Iowa State University and did her master's work in health economics. So with that, I'm going to move into our discussion today. So thank you all for being here with us. So I think I'd like to start. So Dr. Powell covered a lot of ground and a lot of territory, and we have stakeholders who represent all facets of our newborn screening ecosystem here today. So with that in mind, and with in mind the broad reach and broad capacity of our stakeholders who are really interested in understanding how we can ensure that the data that we are collecting is available to inform the decision-making needed by the committee at the time of nomination and review. I think we're going to just dig in a little further to some of the topics that Dr. Powell was talking about. And I think I'm going to start with data collection itself, if that's okay with everybody. Because several times, Dr. Powell, you talked about the assessing the published evidence, and then there was the category of assessing unpublished evidence, and then there's that gray information somewhere in the middle. And so maybe we can talk a little, so one point of clarification is when we talk about that, are we talking just about peer-reviewed journals, or does the committee also consider white papers, posters that have been presented at meetings? Can you talk about what falls into those categories? And then we'll move into maybe a conversation about registries and how we can use some of the powerful tools that the community generates.
KK, do you want to take this one since you've been most directly involved with this?
Sure, absolutely. What the evidence review group, we're well aware that these are rare disorders, and I would say in the last, I don't know, last few years especially, information's been coming out very fast. So with that, we have considered most everything you said, right? Formally published. When we say published, we're referring to things that are in like library databases. So right, part of the standard systematic review process we go through is to run a formal search that's also checked by and run by a medical librarian to make sure that everything, you know, we're including all the sources and keywords that we should be. Then that often includes some conference abstracts or other information, and so those get screened along with the rest. And if it fits the key questions and addresses those, we will absolutely review it. And as far as, let's see, yeah, the definitions are a little tricky, right? So that's kind of, I think, what we consider the published evidence, because they're published in these databases. Then there's this other kind of gray literature, which is very broad. And what that includes is that we go through clinicaltrials.gov. We go through targeted conferences, ones specific, you know, APHL, newborn screening ones. There's other specific ones to different disorders, for instance, like a neurological ones. There were some very specific ones for SMA. And then we will also include, okay, so I think at some point the question came up, we will include any published studies that are, or certainly review them, published studies from Registry Data. But if there's other conference information or other unpublished information that would be relevant, we will consider it. So that's like, it's a pretty wide range. We will include most anything. It needs to have kind of some kind of evidence or data that would be helpful and inform the questions to be able to inform the committee and the decision making. For the gray literature, like things that are in the conference abstracts and the gray literature or the unpublished data that we start to hear about, right, when we hear, oh, there are additional registry data, but we don't have any other analyses on it, on those data, we will work with, during the review process, we will work with folks, if there's time and if whatever the group is, the nomination team or whomever, we will work with them to identify other questions and ways to bring them out.
So let's talk about the registry data a little bit, because I think one of the most powerful tools our patient communities have, and patient communities working with other partners, the clinical trial leaders, clinicians, are really working to collect robust data around our patient communities in a longitudinal way that is not particular to any one clinical trial. So we have some really strong longitudinal data. And for a lot of the patient communities, the therapies that are being pursued in rare disease, maybe therapies that are going through expedited pathways to those approvals, may not have as much clinical data at the time of an FDA approval because it might be based on a surrogate, but there might be other data available from other sources that could help inform decision making. So can we talk a little bit about what types of data might be available in patient registries that would be useful and might not already be being collected, but if we could look at other examples of communities that have gone before, could be data elements that we could be considering adding to existing registries to be collecting this data?
Yeah, sure. You know, and I can actually speak to some specific examples where we've worked with folks and with existing registries. You know, Pompeii was one of the first, and we worked with that group because there was lots of small studies, right, this body of evidence that we had on usually the most challenging part of the evidence review is finding sufficient evidence, or finding any, to be honest, sometimes evidence, published evidence or data about the benefits or outcomes of early detection and treatment. Just because, right, there's not usually a whole lot of screening. So oftentimes there's sibling studies or family studies, things like that. And so, and as much as most of the community is well aware, oh, earlier is better, right? Where you know that, you can see that through case reports, or, you know, you have that feeling, we have to provide evidence to and present that to the committee to consider. So, so one of the things that we did with the Pompeii registry, we actually asked them to run an analysis on, and it's in the report, you can go back to it, of, I think it was of like patients treated before, at less than 12 months and within the first year. And then within that group, we just separated outcomes for those who were treated before 3 months of age and those treated after 3 months of age. And then looked at these broad outcomes, survival and use of ventilator support. And that was it, right? And that was actually really important, helping, right? And it's not super clean, but it was provided another source of information. So with that, right, ages of symptom onset, ages of diagnosis, ages of treatment initiation, ages of onset of critical outcomes, or age, you know, different ages of critical outcomes across time. Those would be some of the main, like, really important ones. If we were to go back, that's where when we've had to go back and talk with a group that has unpublished data, right, that they just haven't actually crunched in a certain way. Those are probably the, you know, some of the important ones. I think with SMA, we also looked at a poster, like that was another kind of critical piece of evidence. We looked at a poster of, which is considered gray literature, kind of a medium light, you know, darker gray, but that looked at disease duration, right? It was based on, you know, coming from a study of all symptomatic infants with SMA, had symptoms less than 6 months, starting at less than 6 months of age. And this investigator had done a poster just looking at outcomes for infants who had disease duration total of like up to 12 months or less versus 12 months or later. So the length of duration of disease symptoms, and just did that cut. And that, again, it also shows it's trying to get at indirectly, is earlier better? Is earlier in the disease progression better? Is earlier in age better?
So this is all incredibly helpful, because I think these are actionable takeaways that people who are in our community can be applying right now.
Yeah.
So one of the things you also said that sort of triggered another thought that we actually all talked about a little bit the other day is siblings. And so sometimes in these registries where we are seeing the earlier, sometimes even a preclinical onset, is younger siblings who are diagnosed. And in rare disease, there are a lot of sibling studies sometimes conducted. There's a sibling arm of a trial. And I'd be interested in your thoughts around, are you seeing sibling data brought forward? Are you seeing any write-ups of these trials? Or is this another area that would be, again, another actionable takeaway for us to think about? So I'd love your thoughts around that. What are we seeing? And what could we be doing about this?
Yeah, sibling data, my sense is that's the first place where clinicians and families are starting to realize earlier is better if we can catch them earlier, right? And so, yes, that's where some of the initial sparks of evidence, if you will, start to come. Whenever there's a published abstract case report of any kind, letter to the editor, you know, that's one of those exceptions. If it's a direct, even a, you know, little paragraph on a sibling, family, you know, the outcomes that have the onset, all those variables again, age of symptom onset, diagnosis, treatment initiation, and outcomes of the different siblings, because presumably, right, the age of diagnosis and treatment initiation start to get earlier with the younger siblings. And so, yes, absolutely, we always report on them. They've been in all the reports. You know, the question becomes, they become an important part of the body of evidence. So the more information we could have on that, the better, right? To be able to, you know, if we had multiple sibling studies, that would be even better.
That's great.
So, yeah.
I've seen, Anna Marie, I've seen you very active in the chat. Thank you very much. One of the things that came up was a question of international data versus US data. Can we just talk, that's a big question. There are a lot of international pilots or countries that are screening. How does that get regarded by the committee? How's that data looked at?
That's KK. Yeah, I was sort of just making sure it didn't get missed, because I know KK would be able to say the review does include international data and literature, of course. Yes.
We will always include it. We will, if there is US data that's similar, that becomes relatively more important. But we will also report any studies, any published studies on newborn screening, all the same thing, early detection, et cetera.
Okay.
For international studies, for sure.
That's great. So I'm going to shift gears just a little bit, and maybe Anna Marie, ask you to maybe start this one off, but everyone I'm sure contribute. But we talked the other day a little bit about a concept that was really important, is this concept of being more deliberate in differentiating cost and impact between the screening ecosystem versus the clinical ecosystem. And I was wondering if you could perhaps reflect on CCHD as an example and talk a little bit more about how you demonstrate preparedness when you think about readiness within the screening ecosystem versus the clinical ecosystem. What data do you need to have ready to demonstrate this? How do you know?
Yeah, I think this one is certainly as challenging as the conversation around looking for, you know, just finding those few case reports, those studies where you found a baby by screening, or getting a decent amount of data, clinical data that's published to put into evidence review. And I saw a couple questions in the chat around PHSI that Dr. Powell touched on. And I think we've had several conversations, both in the committee itself and in the long-term follow-up work group, around that challenge that we have in understanding the difference between a public health system impact of adding a new condition, rolling that out, what that means for the lab, what that means for staffing, what that means for follow-up of those affected families and children, and then what it means, and by follow-up, I mean public health follow-up, right? And I think this is a challenge sometimes when we talk about it, because there's quite a different impact on the clinical ecosystem when we implement screening, and may find more true cases, or just may have more babies picked up or flagged as a positive screen on newborn screening. What does that mean for the specialists and in some cases even primary healthcare clinicians? What's that going to mean for them when we implement something new? And with CCHD screening, I'll just kind of rewind a little bit for those that are newer to this process or are part of the bootcamp are learning, CCHD was only the second screening added to the RUSP that's a non-blood spot screen. So we have hearing screening, newborn hearing screening, and then newborn pulse oximetry screening, which is looking for hidden but asymptomatic heart conditions in babies. And I know one of the things that clinicians were worried about was the possibility of having a number of babies picked up with a pulse oximeter that were hypoxemic, which is what a pulse oximeter does. And we can't just assume, immediately they have congenital heart disease, right? They need a follow-up diagnostic test, which is true of many of the other conditions we screen for. And I think not having a lot of large population health data on screening at that point, there was concern that we'd be referring all this, there'd be like this flood of babies coming in to need echoes or ultrasounds of the heart. And so there'd be a strain on the system to be able to just do those echoes, but then also on pediatric cardiology and radiology to evaluate and assess those echoes. And as it turned out to not be the case, but I remember quite clearly at the time of going through evidence review and when CCHD was advanced to the secretary, that we really didn't have a lot of data to sort of confirm or rule out that suspicion. So all we could do is kind of look at what was out there, and a couple of the larger studies, actually, really the only large studies that had been done were done in Europe. And we were doing one in Minnesota that, and I would show up at these meetings in DC, and we hadn't published yet. We were in the midst of doing this 6-hospital study in Minnesota on feasibility and sort of that false positive, false negative rate for CCHD screening, but I kept like spoon feeding any little bits of data we had. And that's why I made the comment in the chat box too about make sure you're sharing emerging data or studies that are in progress, because it's valuable. It may not be, have the same, be ready for assessment yet, but at least the committee knows, and evidence review would know that they can be watching and expecting that something's coming around the bend. So I'm not sure if that really answered the question exactly, but I guess I think being thoughtful, if you're a nominator or a nominating group, about the impacts not just on the public health system that manages the implementation and rollout of population health screening, but how it's impacting that clinical practice ecosystem if every baby were to be screened for a new condition.
Well, it does answer the question very well, thank you. But it also builds on something Dr. Powell said in her presentation, where Dr. Powell, I think you referred to it as an algorithm for assessing what the impact will potentially be, or what the algorithm for describing the clinical care required. And I'm wondering if maybe we could talk a little bit more about whether there's an example of where this has been done well and how far out that typically goes. So typically when that comes to the committee, are you looking at, short-term, long-term? Are you looking at the state impact? Are you also looking at the impact on the primary care and specialty care provider networks that are going to be needed? So can you talk a little bit more about that? Because that builds nicely off what Anna Marie was just talking about.
Sure. I was primarily thinking about the short-term follow-up needs, ideally to think about the long-term follow-up, but I know that can be very challenging. Just thinking about how many infants might screen positive and need that confirmatory testing. For example, for something like, CMV, and those infants will need, additional workup, and who's going to be providing that, things of that sort. It is, I think there was a comment in the chat about, why would all this information, be needed or be helpful? I think, it's not just the newborn screening lab work, right? It's the whole system that we have to think about, and how would the states be able to handle something like this? And are there experts out there? Or would, for example, a child need to be sent out of state, to access that expertise? For critical congenital heart disease, some children do need to, either be transferred or to, go to where, they can see a expert cardiologist, quickly, but, balancing that with the fact that that could save that child's life, especially if, quick intervention is needed through cardiac catheterization, and so on. So I think it's just, being able to think about that whole process. And, I understand that it can be very challenging to get that data up front. As an example of a very useful algorithm that was published, by New York State with their experience with X-linked adrenal leukodystrophy. And, what were the different steps involved through their pilot study, which I think has helped inform, the committee and states enacting newborn screening for that. And so something like that can be, extremely useful. The other thing I wanted to mention, going back to, the registry information, and that, the evidence review group could look, could go and ask for that registry information. But I mean, ideally, if that information could be gathered and, published ahead of time, to be included with the package, would certainly help expedite the whole process. I know it's hard. I think we talked the other day about, clinicians who are passionate about the value of early identification of infants with a rare disorder, and passionate about, that would be an excellent condition to add to the panel, but they're very busy clinicians, right? There's not enough of them. The committee has been looking at workforce shortages, and they often don't have the time to put that information together themselves. But if, there was, that information could be gathered and put together in like a draft, publication that they then could, look at and add to and things. It's, the looking at a draft is always a lot easier than being the one to actually get that first draft together. Mm-hmm. So that's something where I think all the expertise out there for these rare disorders, if people could, think about possibly doing something like that.
That's great guidance. So one of the questions I wanted to ask came up through the chat, and I think it refers to the slides that you presented, Dr. Powell, was pulling from the slides that you referenced a de-identified screen validation pilot with biochemical genetic confirmation was needed to identify babies as a part of the validation. So the question is, why must a de-identified baby be identified before submitting a nomination?
So I think the idea that, like with GAMP, where, the first time that was submitted and it didn't go through, that, the feedback was that we need to have a pilot study where at least one infant is identified. And I think it was due to the fact that, again, it's not just the laboratory validation, right? It's the whole system. And is there a system in place that would then not only identify the infant, but then lead to confirmation that that infant does have the condition, any, data available about, how they did after having treatment started early. I know that's been controversial about that sort of requirement of having, one case identified, very challenging for these rare conditions. But again, it's because we're looking at the whole system impact and not just a laboratory test validation.
So I think maybe this question refers to the laboratory test validation, because and sorry if I'm misunderstanding the question. I think the question's questioning the de-identification of the baby. And so maybe the question is really focused on the test validation itself. So are those two distinct processes? Maybe you can clarify the needing a validated screen versus then moving that screen into a pilot and then identifying a baby within the pilot. Can we maybe clarify those two steps distinctly?
Well, I think most state labs do use de-identified blood spots when those are available. I mean, they're critical for being able to validate new tests, and also periodically to make sure that, you know, the test being used is valid, accurate. That can happen like when a new piece of equipment is obtained by a lab, you know, they have to go through that validation process, and that's typically done with stored samples, or when it's a new test, you know, often the CDC will provide samples to be utilized. So, you know, it's sort of different things, I think, you know, in terms of the
that's a de-identified sample versus the pilot isn't looking for the pilot. You're looking to identify families who you can then support through the system.
Right. Like some, you know, some states have done, you know, large studies utilizing de-identified dried blood spot samples, you know, which is very helpful, informative, you know, that yes, we can pick the condition up through dried blood spots, but it doesn't really give us, you know, information past that point.
Great. I do want to ask about workforce and capacity issues. It seems we know that there are many opportunities. We have robust therapeutic pipelines in the rare disease space specifically. There are many communities that are working on developing pilots and developing evidence to come before the committee. And we know that we have the technology to be identifying children younger than they're currently being identified, and to be alleviating the diagnostic odysseys that I know that we're all committed to alleviating for the families that we can. Can we talk a little bit about the workforce capacity currently, and then in order to be able to move towards where we need to be in the very near future, if not today, what would be needed?
Well, I think that, you know, it is a major problem, something that the committee has been hearing about for the last 2 or 3 meetings. And, you know, it's something that is troubling. There's many different organizations that are working on this, the different, you know, genetics organizations, and I know other groups like the pediatric endocrinologists and pediatric neurology groups are thinking about it. It is a problem. I mean, there's a shortage of experts in medical genetics and biochemical genetics in the different pediatric subspecialties. You know, people are not going into these fields often because they don't make as much money as other areas do in medicine, especially like where there's, you know, interventions. You know, I think we haven't examined the workforce in pediatric cardiology. I don't know, it's probably something we should think about, but you know, it's a problem. You know, students, after they graduate from medical school, they're often $100,000 or more in debt. And to think about how they're going to pay that back, you know, they often go into the surgical specialties or interventional type specialties, and not into those that are more involved in, you know, areas like treatment for rare diseases. So, you know, we're working on it. We're hoping that, you know, salaries may improve, that there might be loan forgiveness programs, such as, you know, NIH has established for those going into research, but it often doesn't, you know, include people who just want to treat patients, and that's what we need out there. So, you know, at major places in the country, Boston, New York, California, you know, there's often not that much of a shortage, but when we look at, you know, some states where there might be one clinical geneticist in the whole state, and, you know, whereas for newborn screening, you know, those infants have to be seen quickly, but, you know, for standard referrals to genetics, many places have a 2-year waiting list. So, you know, for long-term follow-up care, it is a problem, and something that I think everybody needs to be working on to help alleviate. But it's so unfortunate that, you know, there's wonderful treatments available or becoming available, and who are the experts that are going to be utilizing those and being able to help patients. You know, and there's shortages. I mean, like, I could take more trainees in my medical genetics residency program, but I don't have the funding for them. You know, we've had some wonderful applicants this year, but, you know, I've only got one position that's being funded. So it's very frustrating, you know, so there needs to be more funding for training programs, for fellowship programs in genetics, as well as the other subspecialties.
Great. I have just an overall comment, then I'm going to have a lightning round question for everybody here at the end. I'll tell you what the question is going to be, so you have a second to think about it. So my question to each of you would be, you know, just in hindsight, having seen the nominations that have come through, what would you say is, from each of your purviews, probably the most effective thing you've seen a nominator do to help inform the decision that was made? So something, an aha that you'd have to share with groups that are working on gathering evidence for a nomination that you could pass along to us today. And then just the overall comment I'm going to make is, just on behalf of all of us that are working in this ecosystem, we thank you for all the work you've done to take a step back and look at the evidence review process and look at the additional evidence that may be available to help inform decision making. And it's noteworthy in your presentation that there are a lot of discussions that are still ongoing, and a lot of areas of evidence and data that may yet be included, but we're still trying to figure out how to integrate that information. And some of the comments in this thread have alluded to this, and it occurs to me that some of those seem to be really ripe opportunities to engage with patient communities also, looking at how we measure values, benefit risk trade-offs, outcomes, especially novel outcomes that are being collected. Many of those are things that are work that we're doing in other ecosystems right now with lots of other partners, and would really transfer nicely into this space. And so we have a lot of partners that would be really eager to lean into those discussions as you continue to have them. So thank you for doing this work, and we look forward to continuing it with you. So for the lightning round question, we would just love, you know, we consider it a success when people walk away with actionable items to apply to the work that they're doing. So if you had just a nugget to pass on to another group, what would that be? In addition to all those you've already offered today.
Well, I'll go first. I think, you know, the value of the pilot study is really very critical. And, you know, for example, with GAMT, you know, because they did have the pilot study, a couple of pilot studies, but one in the US that was able to identify a case. I know it's expensive to run those pilot studies. NIH is funding some pilot studies. There's other groups that are, you know, funded to do some pilot studies, but I think the committee worries about, you know, having good conditions that are probably deserving of, you know, evaluation or submission to the committee, where there's just not enough funding for the pilot study because it doesn't have an expensive treatment where a drug company is going to fund that study. So, you know, I think, but that has been, you know, very helpful in terms of providing enough evidence for the committee. I mean, of course, GAMT is currently going through the evidence review process, but it did at least get to that point. So I think that's very helpful.
That's great. Anna Marie.
Yeah, so I wear a few different hats, and I've been honored to be part of this process, both before I was on the committee and now on the committee. But I think putting on my parent hat, I think one of the best things that I've seen done are just, I know this is going to sound a little like cheerleadery, and I don't mean for it to be, but you know, persistence, like keep at it and keep yourself in front of the committee. And one thing I can say, and maybe with a little bit of bias, is that the committee's leadership and also Dr. Kemper and KK, but the DFO for the committee and the staff at HRSA are very responsive to advocates, parents, families, families in coordination with clinical leaders and researchers. And so don't let the perfect, be the enemy of good. If you think even if you think as a parent that you've got, that you've got good evidence, you've got even some personal experience, you have some experience just in trying to gather some data at the institution in which you're primarily, working, because that's where your child is seen. Don't let that be your barrier, right? Just, kind of move forward. And I think what the sort of iterative process that's been allowed for at HRSA is the chance for the team there to provide feedback and fill in and just say, like, this is really good, this is really good, this is where we've got a gap. What do you think can happen here? Because I think I just think as a parent, it can be a little overwhelming when you think about, like, well, we need an NIH sponsored study for it to be anywhere close to being able to nominate a condition. That sounds like a pretty high mountain for a lot of families to climb, frankly. And that doesn't need to be necessarily the mountain to get things going or to feel like you can get things in order, at least to the degree that the conversation around a nomination or submission could happen. So I guess I just want to encourage the families and the amazing clinical advocates that are out there to just kind of keep at it and approach with good questions. I think we're all rowing in the same direction here. And I really thank everyone for even participating in and for Every Life for arranging a meeting like this that allows us to all learn from each other.
Thanks, Anna Marie. That's great advice. Thank you, and great perspective. KK?
Yeah, and mine is kind of similar, builds off of Anna Marie's. I was just thinking, what came to mind was collaboration. I mean, it really needs to be this team, right? And each part is critical of the families, of the clinicians, of the lab folks, the researcher, all of them, and if relevant, the treatment makers. I mean, for good or for bad, big pharma has been helpful in some cases, right? Use it. The resources that have come together across the past decade, the foundations, right, that you were describing have really been amazing, right? If you really just look back. And so I would say, yes, like, use them. And the fact that you're asking these questions to say, what information can we as families, as nominators, be starting to gather, like, to help you is amazing, right? So that because that we want to be able to find the evidence. I mean, we have to do it objectively and systematically, but if it's reasonable, we will absolutely present it. And yeah, so it's just been truly impressive. But I would say now continue to use this great collaborative effort and these organizations that you all have built. And yes, just continue it. Continue the lines of communication. I think it's a much nicer. I at least don't see this as a adversarial type process, or that needs to be a big scary review, right? You got to develop the evidence, and and that's just a matter of fact. And so, yeah, get our input on how to get that.
Well, we thank you for being collaborators and leaders in the space, and for your just unwavering commitment to optimizing the health outcomes of our nation's newborns, and for your generosity of your time here today. So thank you. And with that, I'm going to turn it back to Dylan. And just for everybody, I know there were some unanswered questions. Hopefully we got to most of the themes in the thread, but if there was anything we didn't, we'll try and circle back to that when we post everything from the boot camp. So thank you, Dr. Powell, Dr. Lamb, and Anna Marie for everything today. Dylan?
Great, and thank you guys so much for a wonderful presentation discussion. We're really excited about that, and we're really excited to continue that conversation.